Okay, so we're gonna go ahead and get started, and hopefully this records on my computer. So like I said before, my name is Luani Hernandez. I'm one of the service trip coordinators, and the article that we are going to discuss is Mitofusin 2 regulates neutrophil adhesive migration and the actin cytoskeleton. If you don't know too much about what that is, don't worry, we will kind of explain as we go along. So just to kind of get things started, so what is a journal club? So it's just a general space where we explore various scientific topics. Many things are on the table for exploration. So biological research, biomedical research, you can do a public health article, clinical research, case studies, even if you find like a professional development article, that's another type of research that's often we don't think of, but it's still on the table. You get a chance to present, so I think it's a great opportunity. Not only do you get the chance to kind of learn about how to present, but you also get to explore various topics while you're doing that. It's a great resume builder, just to kind of shameless plug it. I was able to sort of put Journal Club as one of my activities on a resume that got me into an internship. Shameless plug, it was Cornell, so good on you for coming. So today is going to be a little different with time constraints and all that. So typical format, it'll probably be 50 minutes. The general meeting, it's a 30 to 50-minute presentation. The sweet spot is 45. You don't really have to have a set sort of time. It's just kind of like those are the guidelines, and 10 to 30 minutes of questions depending on how much we delve into the article. So today the time is about the same, but I'm just going to do it a lot on content tips. So today I'm presenting, but however, in the future, I'm hoping you guys will volunteer and present. And if you need guidance on that, I am more than happy to help you guys with it. I put a little inspirational quote to motivate you guys to come to Journal Club. So the first thing we're going to discuss is the background for your slides. So this is an example from one of my previous journal clubs that I did for a lab. So kind of looking at it, you can see the schematics between the two. One of them has a fancier background per se, and the other one has a blank background. But in kind of my opinion, and probably yours too, the blank one is much better to see. So blank is sometimes best because it's easier to read. So a lot of times you don't necessarily have to put a super complicated background. Really what you want to be focusing on is your visuals and your content. So of course you have authenticity to your slides, but following the guidelines might make your presentation easier to read. So like you probably noticed, my title slide was very blank. It had the journal club name, the journal club number, the title, and your name. It's optional, but come on, guys, take credit for your work. So this is kind of what you want to start out with. So what you kind of want to start out with is in your top left corner. So that's your general division of content as your introductory slide. So you're going to have acknowledgements and background, gaps in knowledge, goals, your hypothesis, if it's separate from goals. If not, usually you can group that into goals of the paper, and significance. So the best resources for this information are your abstract and your introduction. If you look in the article, it quite literally says abstract, introduction, and it gives you pretty much all this information. You can also consider the titles of your results. A lot of times the results are divided into different sections and they kind of have a title that summarizes what exactly went on within that specific subset of figures. And that's also a great resource. There's a key tip there, and that is to annotate. So annotating just helps you organize your ideas like I did there. I highlighted different things, and I just kind of put like a simple description of why I thought it was important to annotate. So whether it was a summary, a detailed finding, conclusions, honestly, it's up to you, but it just better helps organize your thoughts. So just to kind of get started now on content. So how would I go about using this article? in this format. So the author's acknowledgments were that neutrophils are the most abundant leukocytes in the first lines of host defenses and immunological systems. So mainly they are recruited to inflamed tissues. And what's really important to note here is that the mitochondrial outer membrane protein, Mitofusin 2, so that's abbreviated as MFN2 and will continue to be abbreviated that way throughout the presentation, regulates neutrophil homeostasis and chemotaxis in vivo. So chemotaxis is essentially neutrophil migration. So there's just a basic pathway of neutrophil migration that the article highlights that I thought was important to note. So there's surface receptor activation, which is mainly through G protein coupled receptors. And then you have this cascade of events that results in RAC activation. So you have RAC, which helps with actin polymerization in the cells, which helps drive the migration of neutrophils. So kind of just little surface area here. Actin is essentially a component of the cells that is used for movement. So you're going to see that in a lot of your classes as well, since a lot of you are at the lower levels in biology courses. And what the author denotes here is that mitochondrial function is important for cell migration. When you have a disruption of the membrane, which is the mitochondrial outer membrane in this case, a lot of times you're seeing chemotaxis inhibition. So the membrane is important for things like oxidative phosphorylation that results in ATP intracellular redox regulation and a contribution to calcium distribution. So the main gaps that the author highlights is that they don't know the exact role of mitochondria in neutrophil function. So you know various things about the actin polymerization and how it drives migration, but the exact mechanism remains unknown. And there's still some gaps between RAC activation and exactly how that works in neutrophils. So the goal is just to explore mechanisms outlining the role of mitochondria in neutrophil migration outside of ATP and cellular energy pathways. And the significance that the article highlights is that experiments can contribute to the understanding of MFN2 related mitochondrial disease, as well as better teach us about how we can manage these diseases. So things such as cancer, diabetes, and Alzheimer's. So one thing that you want to include always in your journal club presentations is methods. It doesn't have to be long. I think in the article, it mentions that the methods are about like you see like 2 to 3 pages with detailed components. That's for the purposes of publishing an article. For our purposes, we just kind of want to skim over the brief things that the author did because what's more important, again, is your results. So here they use the zebrafish model. That's for in vivo assays as well as MFN2 knockout mice. Cell culturing in vitro. I don't know if they've discussed this in your classes yet. In vivo usually means within the animal. In vitro is on a cell plate. So just like in vitro fertilization, for example, that's done on a plate. You have confocal imaging, flow adhesion assays, immunostaining, which to me I think is pretty cool when you see the results. It's all the different colors and it looks kind of flashy. RAC GTP pull down assay, so that's a migration assay. And that's microslide chemotaxis, FMLP. That's also a migration assay. So like I mentioned, methods are important, but it shouldn't really be the focus of your presentation. Just highlighting the key ones should be sufficient. So the most important thing is to highlight your results properly. So you want to have a good header. Usually the name of the result header is usually what you use for your presentation. So usually when you find a figure, it will have at the top of it, along with those explanations as to the significance of that figure, like what the main result was. Usually that's what I use. 2 to 3 figures, at most 4. I fit 4 in some of these, but you shouldn't really be crowding it. And you should have a basic descriptor and that you're summarizing the figure and a significance of it. These can be set up however you feel is best for the presentation. And for example, it just might be best to take let's say figure 1 has parts A through J of that figure, you don't have to fit A through J on the slide. Just say, for example, if results 1A and 1C interested you, and you thought were most relevant to the conclusion from that result, you would put that on the slide. But you don't have to highlight everything. It makes for a very brief, concise, and easy to follow presentation. So, kind of to dive into content here. One of the first results that was important to note from this article was that neutrophils depleted of MFN2 accumulated in zebrafish vasculature. So what I kind of wanted to note here in these figures was the imaging and subsequent quantification of MFN2 knockout neutrophil recruitment. So there was in these neutrophils no MFN2 present. And it's really, really clear in the quantification, so figures 1E and 1F, how by knocking out MFN2, there was a loss in neutrophil migration. Figures 1C and 1D are sort of the imaging to support that, but the quantification makes it pretty clear that there's a statistical significance of a difference in between neutrophil migration when MFN2 is knocked out. So just to kind of understand what I'm doing here, if you, for some reason, need help with remembering what you need to say or some sort of script, sometimes people get worried that they may get lost. You can always use speaker notes. I have some of that up right now. I'm not relying too much on it. But if it's one of your first presentations and you feel as though that's really like, I need some guidance, feel free to use that. No one's stopping you. You can also use your phone, note cards, but regardless, you don't want to overcrowd your slide, which is why a lot of people prefer to have subnotes rather than actually putting a whole bunch of words on the slide. It's just to make the figures clearer and easier to read. So another part of the article that I thought was important to mention was that they concluded that MFN2 regulates neutrophil adhesion and migration in vitro and in vivo. So what we're looking at in figures 2B and 2F, they are a little blurry, apologies, but they are looking at the velocity of MFN2 knockout HL60 neutrophil, one is rescue function and one is simply that knockout neutrophil line. So if you note on there, you see SH1 and SH2. SH here is short hairpin RNA, and that is used to basically knock out MFN2. The interesting part here is that HL60 is actually a human neutrophil-like cell. So they are using this, I believe, in zebrafish models, but it is a human neutrophil-like cell. So I think it's pretty cool. What I really thought was cool here was that in figure 2F, we're seeing a rescued function. So essentially, by introducing shRNA resistant MFN2, which is on figure 2F as SH1 plus R, so the rescued function, you're seeing that velocity actually increase again in comparison to the control. So it's more similar to the control, which indicates a rescued function. So figures 2K and N simply show one is that MFN2 deficient cells have hindered adherence to endothelial cells. So the SH1, again, that's knocking out. MFN2 has a statistical significance of a difference in adherence, and it's lower adherence. And figure N actually highlights a mouse breed that was specific for neutrophil specific depletion. And when that MFN2 was knocked out in that line, you can see that the infiltration decreased as well. So you can kind of see how MFN2, by knocking it out and seeing a difference, we're seeing some sort of regulatory component towards neutrophil adhesion and migration. Another thing to highlight here is that MFN2 regulates cell migration through maintaining the mitochondria, ER tether, and DHL60 cells. So MFN2 is localized to both the mitochondria and ER membrane. So ER is endoplasmic reticulum, and essentially regulates the tethering of these organelles. So here we have figures 4E and F shows that MFN2 deficient DHL60 cells exhibit higher levels of cytosolic calcium and a reduction of mitochondrial calcium, which suggests by having these calcium levels have a fluctuation in distribution, it suggests the loss of endoplasmic reticulum and mitochondrial tether is somehow regulated by MFN2. So we have figure F, which shows that they actually here try to essentially create an artificial tether to basically restore the function of these neutrophils in MFN2 deficient DHL60 cells. So we see that the recovery here is in SH1 plus T, that's the tether, the artificial tether. It rescued its chemotaxis, and it suggests a mechanism of MFN2 regulating chemotaxis in DHL60 cells. We also see that MFN2 suppresses RAC activation in DHL60 cells. So here we have figure 6B. So it's showing phosphorylation of PAK. So that is, forget what that is, apologies. But essentially it is phosphorylated, and the phosphorylation is serving as a readout for RAC activation. So by being elevated at multiple time points in comparison to the control, it suggests that there's a suppressive role of MFN2 in RAC activation in DHL60 cells. So we have figure 6H then that I want to focus on here. So excessive phosphorylation of PAK was corrected by expressing the artificial tether in MFN2 deficient DHL60 cells. So you can see again that tether there changes the phosphorylation levels. And that is indicative that MFN2 moderates RAC activation via the mitochondrial endoplasmic reticulum tether again. That's dealing with that sort of mechanism. So then we have figure 7B, and this assay was essentially done to determine whether the heightened activation of RAC is the mechanism leading to the chemotaxis defect in MFN2 deficient cells. So here, MFN2 deficient cells were treated with RAC inhibitors. So just for short, NSC and CAS, those are both highlighted up there. And what they show is that there was a partial restoration of neutrophil migration upon treating with RAC inhibitors. So conclusions and future directions is what I'm getting to next. But it's essentially summarizing the main points. Titles of results help with this, just kind of looking at your results and figuring out what essentially was important from the article. Future directions is also something that a lot of times these journal clubs will ask you to do. And what it is it's basically asking kind of where to go from there. So is it mentioned in the article? Were there any limitations in the study that you think should have been addressed, or the scientists themselves often point them out? If needed, again, you can put your own input into it. So what do you think are some of the limitations of the study? And a lot of times they mention it in the discussion portion of the article. So from here, some conclusions. MFN2 is crucial for neutrophil adhesion and migration. MFN2 regulates actin cytoskeleton and cell migration. Mechanisms of MFN2 regulation of neutrophil adhesive migration include the maintenance of tethers between the mitochondria and the endoplasmic reticulum. And this tether essentially contributes to intracellular calcium signaling, which is important in RAC activation. And the article, while I did not explicitly mention it, they tried to measure both MFN1 and 2, and they saw in the DHL60 cells that only MFN2 is required for migration. Some future directions include exploring whether mitochondrial fission and fusion regulates neutrophil migration, evaluating the role of other mitochondrial shapes regulating genes. You evaluate individual mutations of MFN2 identified in human diseases for their effect on immune cell migration. And another avenue that the authors wanted to explore beyond this study was determine whether decreased mitochondrial membrane potential or reactive oxidative species, I believe it's ROS, contributes to defect in migration seen in MFN2 deletion. With that, I will now get to questions. I can clarify anything that you wanted to sort of see again. I can answer any questions about quite literally everything. So you have complete jurisdiction over your slides and journal topics. Be creative and explore. I don't know if you guys had any thoughts about the presentation, thoughts about the article that you want to kind of discuss now. I know I spoke fast. I don't know. No? All right. This may be an unintentional or intentional plug, but the article actually did come from the research lab I'm currently in, so I think that is exciting. If you are interested in getting into research at Purdue's campus or even in internships outside of Purdue's campus, I've done both. You can talk to me about how to do those, but I think they're a great experience to have as a pre-med. A lot of times we don't focus enough on how to connect the research bench or even the research desk, whether you're doing clinical, whether you're doing wet lab research, to our medicinal advancements that we see on the field. So I think it's pretty cool just to have that background experience. There's obviously MD PhDs and people that do more research with medicine, but if you're just going straight to the clinic, it's still important to have some sort of scientific background over these topics. So the next one, the next journal club is October 2nd, same time, same place. If you are interested in presenting an article, you can email me, group me, DM, kind of expressing interest, first come, first serve. I will approve your article, but I'm open to anything. For everyone's sake, try to get this done by September 25th so I know who's presenting, so I can get it to the newsletter and all those other things so I can communicate exactly that who's presenting. Prepare your presentation and the night before, at least 24 hours before, because we are all people and we are all busy. You can email me the presentation, and if not, if you want to present from your computer, totally fine. We can figure that out too. I just have to figure out a method to record. But there is my email up there. And if you guys don't have any further questions, that concludes Journal Club. I know it was a little short, but like I said in the beginning, it just really depends on how much you have to present and how many questions you have to ask. So yeah, thanks for coming.
Thank you.
Yeah.